The first question that strikes the minds of students is that why we need to control microorganisms. So the simple answer to this question is we need to control microorganisms in order to prevent the transmission of disease, in order to prevent deaths occurring from disease infections caused by microorganisms. So all these are the reasons we need to control microorganisms. Now, the second question that arises in the minds of students is that how to control microorganisms. So we control microorganisms by two methods. One is physical method and another one is chemical method. And this lecture is all about control of microorganisms by physical method. So let's start it. In microbiology, we have a lot of methods by which we can control microorganisms. The first one is high or low temperature. The high or low temperature affects the growth and reproduction of bacteria. It slows down or kills the bacteria. The high temperature kills the bacteria, while the low temperature stops or prevents the reproduction of bacteria. Now, the second method is desiccation. Desiccation means the removal of water from the substance so that bacteria cannot survive in the environment, because bacteria needs water to survive. Bacteria needs bacterial enzymes needs water to work to do their jobs. So now, and the third method is osmotic pressure. If you increase salt in a solution, the solution will become hypertonic, and that hypertonic solution, bacterial cell will behave like other cells, like animal cell, like other plant cells. The water inside the bacteria will be pushed out to the environment, and the bacterial cell will become shrink and ultimately rupture. So in this way we can control microorganism physically. And the fourth method is radiations. Radiation is the method in which we use UV light or X-rays or gamma rays to target DNA of bacteria. If you damage the DNA of bacteria, bacteria cannot do replication, bacteria cannot replicate itself and does not survive. So in this way we can control microorganisms. And the fifth method is filtration. Filtration means physically removing bacteria from juices, from liquids, like you are filtering vaccines, you are filtering polio vaccines, etc. In this way, you are filtering out bacteria physically. Now, we are moving toward the physical methods in detail. The difference between moist heat and dry heat is that moist heat uses water molecules to coagulate the proteins of bacteria, and dry heat uses oxidation to destroy the DNA of bacteria. Dry heat also produces free radicals, which are very reactive molecules, and ultimately destroy the DNA of bacteria. Autoclaving is a common example of moist heat. It operates at temperature typically at 121 degrees centigrade for 15 to 20 minutes at 776 mm of Hg. In this condition, all microorganisms plus spores are eliminated, destroyed, and killed. So autoclaving is a method of complete destruction of microorganism and its spores. The dry heat uses the temperature between 160 to 170 degrees centigrade for 2 hours, and this way you can remove bacteria from their substance. Dry heat is mainly used for powders, oils, and glassware because they are damaged by moist heat. If you provide water to the powders will be damaged. Now, boiling. Boiling is another physical method used to kill most of vegetative bacteria, not spores. Spores are not killed by boiling. So we boil milk, water, or other liquid substance for 100 degrees centigrade for 10 minutes, and it will wipe out vegetative bacteria and viruses. So boiling is a disinfection, not sterilization, not complete destruction of spores and microorganisms. Bacteria like Clostridium and Bacillus will still remain alive in boiling waters because they are very resistant to heat. Sterilization was named after Louis Pasteur. This method uses controlled heat at lower temperature for 72 degrees centigrade for 15 seconds to kill pathogens in food and beverages without destroying taste or nutrients. For example, we pasteurize milk, fruit juices. This kill organisms like Mycobacterium, tuberculosis, Salmonella, Listeria, but again, it does not kill heat-resistant bacteria and spores like Clostridium and Bacillus species, which are very resistant to the heat because of its thick cell wall. Now, heading toward incineration. Incineration simply means complete burning, used to destroy infectious waste, use syringes, contaminated dressing, and laboratory cultures. All these are incinerated at temperature 500 degrees centigrade to 1000 degrees centigrade temperature. Now, cold and low temperature. Cold temperature actually and basically it slows down the bacterial growth. It only stops or slow down the bacterial enzymes from replication, from production, and from metabolism. So refrigerator at 4 degrees centigrade keeps food safe for days. Freezing at minus 20 degrees centigrade or below can preserve samples for years without killing bacteria. Now heading toward radiations, we use two types of radiations to kill and damage the DNA of microorganisms like UV light, X-rays and gamma rays. As we know that UV light has the wavelength up to 60 nanometer which targets the DNA and damages the DNA. In this way, bacteria cannot divide and replicate, so it is eliminated and wipe out. Mostly we use UV light to disinfect operating theaters, air, biosafety cabinets and water purification. What is the limitation of UV light? UV light cannot penetrate glass or plastic. It means it is only used for surfaces or air but not for the instrument that are inside in the box, like medical instrument or like other packages material. We cannot use UV light for that purposes. And the next one is ionizing radiation, which includes gamma radiations and X-rays. X-rays and gamma radiation are quite powerful radiations. They can penetrate deeply into the packages, into the glasses, and destroy the organism, the microorganisms. So it is mainly used for disposable syringes, catheters, gloves without opening the packages. Almost all disposable medical equipments in hospital is sterilized using gamma radiation at the factory before it reaches. Now coming toward the filtration. Filtration physically removes microorganisms by passing a liquid or gas through a filter with pores small enough to trap them. We have many types of filter, for example, HEPA filters, which are high efficiency particulate air filters in hospital, which filters out 99.97% of airborne particles. It is used in operating theaters, ICUs and isolation rooms. For example, if there is a patient of coronavirus, if there is a patient of TB, they are isolated in rooms, and in that rooms we have installed HEPA filters so that airs from that room cannot move out to the environment and do not cause disease in other healthy population. We have membrane filters with a pore size of 0.22 microns, which can filter out even the smallest bacteria, because bacteria are in the size range of 0.2 micrometer to 10 micrometers. So in this way, we can filter out the smallest bacteria possible. Now, viruses cannot be filtered because their sizes are very small as compared to the bacteria. Their sizes are 10 to 300 nanometer. We use filtration to sterilize liquid that would be destroyed by heat, like certain antibiotic, vaccines and eye drops.